Hi, Dr. Finn.
Hi, how's it going?
Good. I've had only 2 people come in and join in, but I'm just waiting for more people.
All right. I was visiting Estefania earlier. She didn't have anyone until now, and then I left when someone else joined.
Yeah, there were 2 people, but both of them were, I knew both of them. They were just there to support me, so I just presented my poster and then they left after that.
Cool.
Yeah, but I haven't had many people.
All right. You want to show me your 2-minute version?
Yes.
All right, let me close my door.
Hi, my name is Riyasa. My research is in inorganic chemistry for synthesis and characterization of new ball type thiocyanines Thiocyanines are macromolecule compounds, and traditionally they were used used as colorants, as blue and green pigments for automotive paints and printing inks, and as blue and cyan dyes for textiles and papers. The ball-type thiocyanines were reported fairly. They were reported around early 2000s. So they were very new, and there's a lot of interest in research for the ball-type thiocyanines. In my research, the way I synthesized ball-type thiocyanines, there is a two-step process to synthesize that. And the first process is to make the monomer, and the second is to use the monomer and a type of metal to make the ball-type thiocyanine. Under synthesis, we can see the first step is to make the monomer using the 4-nitrothiolonitrile and an octafluoro compound, and the potassium carbonate and acetonitrile in acid reflux condensation for approximately 24 hours to make the monomer. And the second reaction shows us the use of monomer and the copper chloride to make the ball-type thiocyanine in a microwave reactor. And the third reaction shows us the use of monomer and cobalt chloride to make the ball-type thiocyanine in the microwave reactor. The color that I obtained from these ball-type thiocyanines were dark green color. So that is the primary focus of the research. That is the color that we want. And the way we characterize these ball-type thiocyanines from these reactions, we're using IR and UV-vis spectroscopy. Under characterization, we can, in figure 2, we can see the comparison of the IR spectrum of the ball-type thiocyanine with cobalt chloride, which is in purple, and the monomer, which is in red. The main primary focus for us to see is how much monomer has been consumed during the reaction, and that peak can be seen in 2235, which is the peak for the C2N triple bond in the monomer. So we want that peak to be consumed, and we want to see no peak at all or a very less intensity in the ball-type thiocyanine, which is in purple here. And we can see that there is a small peak. That means the ball-type thiocyanine that we obtained from the experiment was not as pure, but it is getting pure as we use TLC and separation to purify the ball-type thiocyanines. And the third figure shows us the UV visible spectroscopy of ball-type thiocyanines with 3 different metals. We can see the locations of the 2 peaks are very similar. This is because of the general structure of ball-type thiocyanine, which can be seen in the result of the reaction. It is only because of the type of metal used that can shift the peaks, and also the intensity of the metals. Intensity of the peaks is because of the purity and concentration of the thiocyanines used for the UV visible spectroscopy. For future work, we will be purifying more of our ball-type thiocyanine compounds and comparing it with the and doing different methods of characterization, such as IR, NMR, UV-vis spectroscopy, and comparing it with the monomer and the past results to see how much of the monomer has been consumed in the reaction. And we'll also be making a single crystal to make sure the structure that we got is correct. And I would like to acknowledge my mentor, Dr. Esther Truffin, Scholars Academy, and the Wells Foundation for making this research possible. It is not 2 minutes, is it?
It's a little longer, but it's okay. We have a visitor.
Oh, yeah.
Hey, it's been forever.
Yes, I know.
Hi, everyone. My name is Riyasa, and my research is in inorganic chemistry for synthesis and characterization of new ball-type thiocyanines. Thiocyanines are macromolecule compounds with optical, electronic, catalytic, and structural properties. and they are the second most important class of colorant. Traditional uses of phthalocyanine colorants are for automotive paints and for printing inks and as blue cyan dyes for textiles and papers. In the visible region in the electromagnetic spectrum, phthalocyanines are limited to blue, cyan, and green colors. The ball-type phthalocyanines that I obtained from my lab was dark green in color, so that is the main primary color that we will see in my research. And phthalocyanines have a wide range of applications in biomedical and material sciences. For example, in photodynamic therapy, primarily for skin cancer. And we also see applications in chemical sensors, optical data storage, reverse saturable absorbers, solar screens, laser techniques, sensor for gases, and many other applications. Ball-type phthalocyanine specifically is fairly new, and there is a lot of research available, research going on for ball-type phthalocyanines. And the way I synthesized ball-type phthalocyanines in my lab was there was a two-step process. The first was to make the monomer, and the second was to make the ball-type phthalocyanine using the monomer and a type of metal. And as you can see, the first reaction under synthesis is the synthesis of monomer using 4-nitrophthalonitrile and octafluoro compound. The monomer that I obtained was white in color. And the second synthesis is the use of monomer and a type of metal, which is copper chloride, in a microwave reactor to get the ball-type phthalocyanine. And the third reaction is the use of monomer and cobalt chloride to get the ball-type phthalocyanine. The colors for these ball-type phthalocyanines were dark green in color, and the way we characterize these, we're using IR. And for figure 2 under characterization, that is the IR for ball-type phthalocyanine with cobalt chloride, which is in purple, and the monomer, which is in red. The main primary peak that we focused on was the 2235 peak shown here, which is the peak for C2N triple bond shown in the monomer. So mainly when we take IRs, we are looking for the consumption of the monomer. So when we get our ball-type phthalocyanine, we will see if all the monomer has been consumed or not. And so the 2235 peak will not show at all. But as we can see in the purple line, which is our ball type, there is a small monomer peak. It's not as intense, but there is still monomer in the ball type. So in order to purify this, we used TLC and separation, and then took the IR again. This is a repetitive process until we actually see the consumption of all the monomers. The other characterization technique we used was the UV visible spectroscopy, which can be seen in figure 3. This is for the ball-type phthalocyanine for 3 different metals. We can see that the location for the 2 peaks are very similar. This is because of the general structure of ball-type phthalocyanine, which can be seen in these reactions. And basically, UV-Vis shows us how UV-Vis is also used to see how pure our concentration is, how pure our ball-type phthalocyanine is. And we know the location is the location is the same because of the general structure. It's just because of the metals, there is a slight shift in our graph. And for future, we will be doing more purification and doing more characterization using IR, UV-Vis, and NMR to check how pure our compound is. Also, we'll try to grow a single crystal to make sure the structure that we got in the reaction is correct for my research. And for the people who joined in later on, phthalocyanines are compounds, macromolecule compounds that are the second most important class of colorant. They're usually used in printing inks and in textiles and papers, but this specific new ball-type phthalocyanines, they have found a wide range of applications in biomedical and material sciences. specifically for photodynamic therapy because they are a lot more stable than and a lot less harmful than the other types of photodynamic therapy, specifically for skin cancer. Are there any questions?
Yeah, I have a question. Wait, other than today, how many times have you presented this project?
This is my first time presenting this project.
Okay, so my real question is like, were you like ever nervous about having to do like all this research, making sure you had it right and like, possibility of failure and all?
Yes. So the first time I joined this research, I had a hard time figuring out what I actually was doing. And it was scary because no matter how much I did these experiments, I would not get any results. So that was very scary. But talking with Dr. Trufen, my mentor, it did help a lot. And she guided me through every single one of that. And it does get frustrating because you have to repeat the experiments over and over again since I'm purifying my compounds. And if I don't see the results, I do get dejected and I do get, it's upsetting, but I guess the more you do it, the more you get used to it. And it's just a matter of, it just takes time for you to get the results.
Thank you.
Mm-hmm.
I lost what was the peak for the monomer that you can see in the IR spectrum?
Yes, you can see the monomer on figure 2. The peak is the 2235. Can you see my cursor? Can you see my arrow?
Yes.
So that is the peak that we're seeing. That is the peak for the C2N triple bond here in the monomer. So in the reaction, that bond will be consumed and C2N double bond will show up. And these peaks over here in purple, that's the emergence of the C2N double bond, which can also be seen in the ball type here.
Mhm.
So basically, we're looking for the reduction of the peak, this peak, and then emergence of more of these peaks.
I see. Thank you.
Mm-hmm. Are there any more questions? Okay, thank you so much for listening. I think there were some people who just joined, so I'll go over my presentation again. So hi everyone, my research is in synthesis and characterization of new ball type phthalocyanine. And phthalocyanines are macromolecule compounds with optical, electric, catalytic, and structural properties. And they're mostly used in colorant and in printing inks and sign dyes for textiles and paper. And in the visible region in the electromagnetic spectrum, phthalocyanines are limited to blue, cyan, and green colors. And in my research, primarily the phthalocyanines that I got were dark green in color. So that is the color that I've been focusing on. There are wide ranges for use of phthalocyanines in biomedical and material sciences. For example, for photodynamic therapy, specifically for skin cancer. And it also, and their other applications are such as chemical sensors, optical data storage, sensor for gases, laser techniques, solar screens, etc. And ball type phthalocyanines specifically are fairly new. And there has not been a lot of research on these because they were found around beginning of 2000s. That's why there was a lot of interest in the research. The way I synthesized the ball type phthalocyanines in my research was a two-step process. The first was to make the monomer, and the second was to use the monomer to make the ball type phthalocyanine. And under synthesis, we can see the first reaction is to make the monomer, and the second is to use the monomer to make use the monomer and the metal copper chloride to make the ball type phthalocyanine. The third is to use of the monomer and the cobalt chloride to make the ball-type thiocyanins. These were done in the microwave reactor. In order to characterize these ball-type thiocyanins, I used IR and UV vis spectroscopy. And under characterization, figure 2 shows us the comparison of the ball-type thiocyanin that I got with the metal cobalt chloride, which is in purple, and the monomer, which is in red. So the main primary focus is the peak at 2235, which is, I labeled it as a monomer peak, but that is a peak for C2N triple bond, which shows up in the monomer. So basically, we want the monomer to be consumed during the reaction, and therefore, in the purple peak for the ball type, the monomer peak will not show up at all, or it will be, or there will be a very less intense peak left in the IR, which can be seen here. And in order to purify this and see no peak at all, we use TLC and separation to purify our compounds as much as possible, and used IR to characterize it again and compare it with the monomer. And the third figure shows us the UV visible spectroscopy of the 3 different metals used. The zinc acetate was used by a past student. I used the copper chloride and the cobalt chloride to make my ball-type thiocyanins. And we can see the location of the peaks are very similar. These 2 peaks, the locations are very similar because of the general structure of thiocyanins. As we can see in the reaction, the structures, it's very similar. It's just the type, it's just because of the type of metal, there's a slight shift in the peaks. And also the intensity of these peaks depend on the concentration that was put in the UV vis spectroscopy machine. And for future, we will be purifying these compounds more and more and using different methods of characterization. And also we'll try to grow a single crystal so that we can check if the compound that we got has the correct structure for the ball-type thiocyanin. Do you have any questions? No? Okay. Hi, Elizabeth. I'm gonna explain my research to Elizabeth again. My research is in inorganic chemistry for synthesis and characterization of new ball-type thiocyanins. Thiocyanins are macromolecule compounds, and they were traditionally used for automotive paints and printing inks as blue sign dyes for textile and paper. And in the visible region, the thiocyanins are limited to blue sign and green colors. The ball-type thiocyanins that I got were very dark green in color. And thiocyanins have a wide range of applications in biomedical and material sciences, specifically for photodynamic therapy and for skin cancer, and also for chemical sensors, optical data storage, reverse saturable absorbers, solar screens, laser techniques, etc. And thiocyanins also have high thermal stability, high thermal and chemical stability, which makes them a lot less harmful, and they can also stay for longer. And ball-type thiocyanins specifically are fairly new. Therefore, there is a lot of research interest in them. And the way I the way I synthesized ball-type thiocyanins were as a 2-step process. The first step was to make the monomer, which can be seen under synthesis. The first reaction is the making of monomer in a reflux condensation reaction. And then the second step is to use the monomer and a type of metal to make the ball-type thiocyanin. And then the third is also the use of the monomer and a different type of metal. So the metals that I used was copper chloride and cobalt chloride. These reactions were done in a microwave reactor. And after I obtained them, they were very dark green in color. And in order to characterize them, I used IR spectrum and IR spectrum and UV vis spectroscopy. And under characterization, we can see for figure 2, there's a comparison of IR spectrum of the ball type phthalocyanine that I got from cobalt chloride, and the monomer is in red and the cobalt chloride is in purple. The main peak that we're looking at is the 2235 peak, which is the peak for C2 and triple bond, which is shown in the monomer. So we basically want the peak to be consumed in the reaction. Therefore, for the ball type phthalocyanine, which is in purple, we don't want that peak at all. However, in my sample, there is a small peak for that specific bond. That means the sample is not as pure. And in order to purify this sample, I used TLC and separation with TLC to purify my sample. It's a very repetitive process. I'm still at the process of purifying this sample. However, this is the most pure that I have gotten. And the third figure shows the UV visible spectroscopy of polymers with different metals. So the two metals that I used was copper chloride and cobalt chloride. The one in purple is with zinc acetate from past student. We can see the location of these are very similar. That is because of the general ball type phthalocyanine structure that is seen. It's just because of the different types of metal use. That's the reason why there is a slight shift in these peaks, and also the intensity depends on the concentration and the purity of the sample. And in future, I'm still purifying my compounds. So in future, I'll be purifying more, doing more TLC and doing more IR to characterize them, and also use other methods of characterization. And also I will try to grow a single crystal to make sure the structure that I've gotten from this specific reaction is correct. And that is basically my research. Do you have any questions?
So when you worked with both different metals, which do you had one that was easier to work with?
So when I was working with these metals, it was hard for me to dissolve these in just acetone, since acetone is the main primary solvent that I've been using. Mm-hmm. So there were 2 fractions of each metals. I used acetone and DMF for both of them. And for me, cobalt chloride was more easy to work with because I purified cobalt chloride more. As you saw under characterization, the more pure is for the cobalt chloride that I got. So I think cobalt chloride was a lot more easier for me to work with. And also with copper chloride, when I was purifying it, for some reason, the color changed to bluish in color, but I need it to be dark green. So I'm still working with that and figuring out why it changed, why the color changed.
And then you said for the purity, you had to do TLC for that?
Yes, so we use the big TLC, and what we basically do is we put our sample at the bottom, make a line, and then let it sit in our solvent, and then and after all our sample, after we see the sample being moved up, we check under the UV light, and we see a lot of clearless stuff moving up, and some of the monomer that had not been reacted also move up. And we extract only the dark green compound because we know that's the only phthalocyanine that we get, and that's how we keep on purifying the phthalocyanines.
That's cool. And I was wondering, so you said it worked also for photodynamic therapy. Yes. So how would they use it for that, if you know?
So there is still research been going on, but I do know that because of the thermal and chemical stability of phthalocyanines, it's a lot less harmful used for skin cancer. So the way photodynamic therapy works is there is a photosensitizing agent that is put on your skin, and when it reacts with light, it starts killing the cancer cells because there's a specific type of oxygen molecule that gets formed, and then that starts killing the cells. And it also alerts the immune system to attack the cancer. So the photosensitizing agent that had been used is a lot harmful. But if you use phthalocyanine as that photosensitizing agent, it's the side effects for that specific treatment is not as much. But there is still more research going on that.
Thank you for answering that.
You're welcome. Thank you so much for listening. If there are any more questions, please feel free to ask.
So I'm taking Bio 2 right now. So would you recommend taking genetics first or the microbiology?
I haven't taken microbiology because I'm a chemistry major, but my friends, they take genetics first before taking microbiology. I'm not sure if that is the right way, but I'm taking genetics right now and I really enjoy it. And I think microbiology is a very heavy content-based course. So, and genetics, it is heavy too, but I think it's very interesting in my sense. And if you take it with Dr. Kang, it will be a lot more understandable.
Right, Dr. King. But
Yeah.
I don't see Dr. Kang for the fall semester. I only see Dr. Menard and Dr. Wilson.
Yes, I don't think Dr. Kang teaches during the summer. She does teaches during the fall. I suggest taking Dr. Kang because I haven't taken other professors yet, but I do like how Dr. Kang teaches. It's very interactive, and also the labs are very interesting. So if you can take it during the fall with her, that would be great. But I do have friends who are taking genetics during the summer.
Right.
So you'll probably see them, and I can introduce you to them. You can have study sessions together if you want.
But I'm actually taking organic 2 in summer, so organic 2 is in the same time as genetics, so I can't take genetics in summers.
Okay, you should take it during the fall then. Is Dr. Kang not teaching during the fall?
No, she's not.
Oh,
It's
that's
Dr. Wilson and Dr. Menard only. They were the only 2 professors I found.
I took Dr. Menard for general bio, and she was okay, I guess. But yeah, that is really bad because Dr. Kang is like the best for genetics.
Got it.
So if you can wait for the other semesters, that will be okay, you know?
Makes sense.
Yeah.
And for bio, have you taken biochemistry?
No, I'm taking it during the summer.
Ooh, okay. So please let me know how it goes. And what professor do you have?
For biochem for the summer?
Yes.
I have just the biochemistry lecture and lab.
Okay. All right. Thank you so much.
You're welcome.
Take care.
Thank you for coming.
Mm-hmm.
Hi, everyone who just came in. My name is Riyasa, and my research is in inorganic chemistry for the synthesis and characterization of new ball-type thiocyanines. Thiocyanines are macromolecule compounds with optical, electronic, catalytic, and structural properties, and they're the second most important class of colorant. And specifically, copper thiocyanines, it is the largest volume of colorant sold. And traditional uses of thiocyanine colorants were for automotive paints and printing inks, and as blue sign dyes for textile and paper. In the visible region for the electromagnetic spectrum, thiocyanines are limited to blue sign and green colors. In my research, the ball-type thiocyanines that I got were very dark green in color. So that is the main primary focus of me. Whenever I'm separating and purifying my compounds, I look for the dark green color. And thiocyanines have a wide range of applications in biomedical and material sciences. For example, in photodynamic therapy, chemical sensors, optical data storage, reverse satural absorbers, solar screens, laser techniques, etc. And thiocyanines have high thermal and chemical stability. That is the reason why they're more suitable for photodynamic therapy for skin cancer specifically. Ball-type thiocyanines specifically are fairly new. They were discovered around in the early 2000s, so there's a lot of interest in this specific compound. And the way I synthesized ball-type thiocyanine it was in a two-step process. The first was to make the monomer, and the second was to make the ball-type thiocyanine, which is the polymer. And under synthesis, we can see the first reaction is for the making of the monomer, using an octafluoro compound and 4-nitrothalonitrile in a reflux condensation reaction for about 24 hours to get the monomer. And the second step was to use the monomer and a type of metal to get the ball-type thiocyanine. We can see in this reaction there is the use of monomer and the copper chloride in a microwave reactor to make the ball-type thiocyanine. And the third reaction was use of monomer and cobalt chloride to make the ball-type thiocyanine. As I said before, these compounds were very dark green in color. And the way I characterized them was using IR spectrum and UV visible spectroscopy. Under characterization, figure 2, you can see the comparison of the IR spectrum of the ball-type thiocyanine with cobalt chloride, which is in purple, and the monomer, which is in red. So the main peaks that we're looking at is the 2235 peak over here, which is the peak for C2 and triple bond in the monomer. So primarily, we're looking for the consumption of all the monomer because we want our sample to be as pure as possible. So therefore, this 2235 peak will not be there at all for the ball-type thiocyanine. However, it is a little hard to purify these compounds. That's why we need to go through different steps of separation using TLC to purify these compounds. The most pure I've gotten is in figure 2 for the cobalt chloride ball type. As you can see, the peak for 2235 for the purple line, it's very small, and I'm still in the process of purifying it so that I don't see that peak at all. Another thing that we look for the IR is the presence of these peaks here, which is the emergence of the C2N double bond in the ball type thiocyanine, which can be seen in the structure itself. So that's how we characterize if we have gotten the correct structure or not. And the third figure shows us the UV visible spectroscopy of 3 different metals of ball-type thiocyanines. The blue one is with the copper chloride, and the pink one is with the cobalt chloride, and the purple is with the zinc acetate. Zinc acetate was from a past student. And as I compared them, we can see the 2 peaks here, they are in the same similar location. This is because of the general structure of ball-type thiocyanine, which can be seen here. It's just because of the different types of metals used. which shifts the peaks a little. And also the intensity of the peaks are because of the purity and concentration of the phthalocyanine samples that I got from my experiments. And for future, I will be purifying these compounds more using TLC, and I will also be using different methods of characterizations other than IR and UV. And also after I purify these compounds, I will try to grow a single crystal to make sure the structure that I got for my sample is correct. That is an overview of my research. Are there any questions? Thank you for listening.
Oh, that was my question. I was typing it, Riyasa. Sorry.
Oh, I didn't see it. Okay, hold on. Let me see it. So the most interesting thing I came across was, I guess this is my first time using an IR because I was not able to use the IR machine for organic chemistry one. So using the IR and checking if the monomer has been consumed or not, and just repeating the TLC and separation process, that was the most interesting thing. And also another interesting thing that I came across was the sample with copper chloride. For some reason, the more we purified it, the more it turned bluish greenish from very dark green color. So we still need to figure out why it turned bluish greenish, but that was very interesting to see.
Thank you.
Mhm.
I mean, the bluish greenish thing, it's very interesting. And you're
Yeah,
like, well, what's happening here?
yeah. Thank you so much for listening.
So what were all the techniques that you used? Like you mentioned IR, right? And then another one I can't remember.
Yes, IR, and then we use the microwave reactor for the synthesis of ball-type phthalocyanine, and we also use the UV visible spectroscopy. We did do one NMR, but I could not put it in here because it's not it was not the purified sample. However, I'm planning to use the NMR for future work. And I also use the reflux condensation reaction to make the monomer.
Oh, okay. Those were fun. All right. Thank you.
You're welcome. Thank you so much for listening. Thank you for coming.
Yeah. Great presentation. Love it.
Thank you. If you have any more questions, you can type it in the chat.
I guess the multiple repetitions are really helping.
Yes, I do need water because I've been repeating the same thing over and over again. But I'm getting really good questions. That's actually good. Yeah.
It's really awesome. What was your most interesting question so far?
So they asked about when I was trying to do under 2 minutes, they asked about more about the peaks and how they associated with the different bonds. So I explained them to them. That was actually fun.
All right. I'll be back a little bit later.
Okay. Hi, Araceli.
Hi, how are you?
I'm good. How are you?
Good.
So as you know, my research is in inorganic chemistry for the synthesis and characterization of new ball-type thiocyanines. And thiocyanines are macromolecule compounds with optical, electronic, catalytic, and structural properties. And they're the second most important class of colorant. Traditional uses of thiocyanine colorants are as blue and green pigments for automotive paints and printing inks, and as blue cyan dyes for textiles and papers. And in the visible region in the electromagnetic spectrum, thiocyanines are limited to blue, cyan, and green colors. The primary color that I saw in my samples were dark green in color. So that's the color that I've been looking for whenever I separate them and purify them. And thiocyanines have a wide range of applications in biomedical and medical sciences. For example, in photodynamic therapy, specifically for skin cancer, and for chemical sensors, optical data storage, reverse saturable absorbers, solar screens, laser techniques, and many other applications. Ball-type thiocyanines specifically, they were found fairly recently, actually early 2000s, and therefore there's a lot of different research interest in ball-type thiocyanines because they're a lot more stable thermally and chemically, specifically for photodynamic therapy. And in my lab, I used two-step process to make the ball-type thiocyanine. The first one was to make the monomer, which can be seen under synthesis. The first reaction was to make the monomer, and the second reaction was to use the monomer and a metal, which is copper chloride, to make the ball-type thiocyanine. And then the third reaction was to use the monomer and use another type of metal, which I used was cobalt chloride, to make the ball-type thiocyanine. And as I mentioned, these ball-type thiocyanines were dark green in color. And in order to characterize them, I used IR and UV-Vis spectroscopy. Under characterization for figure 2, you can see the comparison of the ball-type thiocyanine for cobalt chloride in purple, and then the monomer, which is in red. So when we characterize these and check if we got the correct structure or not, we look for the main peak that we looked for was the 2235 peak over here, which is the peak for C2N triple bond, which is in the monomer. So primarily, we're looking for the consumption of monomer so that we don't see a peak at all for in the ball-type thiocyanine, which is the purple. This sample is the most pure sample I've ever gotten because the 2235 peak for the ball-type thiocyanine is not very intense. And eventually, as I purify it more, I don't want to see that peak at all. And the other peaks that we want to see in the IR are these peaks that emerge over here. These are the peaks for the C2N double bond that emerges when the reaction is complete, which can be seen in the ball-type thiocyanine over here. And the way I purified my samples were using TLC and separating these samples, and then again using the IR to compare them with the monomer and the sample before. And figure 3 shows us the UV visible spectroscopy of 3 different metals. The blue one is the copper chloride, the pink one is the cobalt chloride, and purple one is the zinc acetate. So these These zinc acetate was from a past student, but as we can see, the two peaks have similar locations. That is because of the general structure of thiocyanine, which can be seen here. It's just because of the different metal that's used. That's why there's a slight shift, but the location is known because of the general structure. And the intensity of these peaks depends on the purity and concentration of the ball-type thiocyanine that was used for the spectrum. For future, I will be purifying my sample, more, and trying to reduce the monomer and consume the monomer. And I'll also try to grow a single crystal to check if this structure that I got is correct or not. And that is the overview of my research. Thank you for listening. Do you have any questions, Araceli?
Wow, that was a lot. I think this is a really great presentation. Like the research itself is very interesting and the way you presented it was great.
Yeah.
What was a challenge you came across?
So just purifying the compound, as I said, I need to not see that 2235 peak at all. And just the repetition of doing TLC and purifying the compounds, it can get very frustrating if I don't see any results. But eventually, I did see some results, and hopefully I will see better results in the future.
Nice, nice. Well, thank you. Thank you so much for this information.
Thank you so much for listening. Thank you for coming. Hi, Jessica.
Hi, Riyasha. I'm a little late.
No, it's all right. I'll go over again. So my research is in organic chemistry, as you know, for the synthesis and characterization of new ball-type thiocyanines. So thiocyanines, as an overview, is are macromolecule compounds with optical, electronic, catalytic, and structural properties. They were traditionally used as colorants, as blue and green pigments for automotive paints and printing inks, and as blue cyandiles for textile and papers. And in the visible region in the electromagnetic spectrum, thiocyanines are limited to blue cyan and green colors. And my ball-type thiocyanine was dark green in color specifically, so that's the primary focus of the whole research. And thiocyanines have a wide range of applications in biomedical and medical material sciences, for example, in photodynamic therapy, specifically for skin cancer, and in chemical sensors, optical data storage, reverse saturable absorbers, solar screen, laser techniques, and many other applications. Ball-type thiocyanines are fairly new. They were found in the early 2000s. So there is a lot of interest and research for ball-type thiocyanines because they are thermally and chemically stable, unlike the other compounds, specifically for photodynamic therapy. And the way I synthesized my ball-type thiocyanine in my lab was a two-step process. The first one is to make a monomer, and the second step is to use the monomer and a type of metal to make the ball-type thiocyanine, which is the polymer. And under synthesis, we can see the first reaction is the reaction to make the monomer, which was done in a reflux condensation reaction for approximately 24 hours. And the second reaction was using the monomer and the copper chloride metal to in a microwave reactor to make the ball-type thiocyanine. And the third reaction was using the monomer and cobalt chloride in the microwave reactor to make the ball-type thiocyanine. As you can see, the general structure of these are very similar. It's just the metal that resides in between these complexes are different. And in order to characterize these, I used IR spectrum and UV visible spectroscopy. And under characterization for figure 2, there is a comparison of IR spectrum of the ball-type thiocyanine that I got from cobalt chloride, which is in purple, and the monomer, which is in red. So the main peak that we look for is the 2235 peak, which is the peak for C2 and triple bond, which is in the monomer. So essentially, we want the monomer to be consumed in the reaction, and therefore this peak should not be in the ball type phthalocyanine at all. It is difficult to purify this compound, so it does take a lot of patience and a lot of TLCs to purify the compound, so that I don't see any peaks for 2235 at all. The most pure I've gotten is for this sample in figure 2. As you can see, the 2235 peak is not as intense. It's very little, and I'm still in the process of purifying it more. Other peaks that we look for is the emergence of peaks over here in purple. Those are the peaks for C2N double bond, which can be seen in the new ball type phthalocyanines. So these peaks tells us that we are getting the correct sample and correct structure. And in order to purify these, we use TLC and separation, and then take the IR again to characterize the samples. The third figure shows us the UV visible spectroscopy of 3 different metals, copper chloride, cobalt chloride, and zinc acetate. As we can see, the locations of these peaks are similar because of the general structure of phthalocyanines. It's just because of the metals that are used during the reaction that causes the shift. And also the intensity depends on the concentration and purity of the samples. And in the future, I will be doing more TLC and doing more purification, and try to grow a single crystal to make sure the structure that I got is correct for the ball type phthalocyanine. Do you have any questions?
Yeah, so what are some challenges that you faced during the synthesis portion?
So before starting off with the metals, I tried synthesis with other metals. I think I tried with manganese, and it did not go well. The color that we're looking for in the lab is dark green, because phthalocyanines are supposed to be dark green. And just the synthesis part was not as hard, because we're just getting the compound, and if it's dark green, then we can work with it further. The main part is the purification of the sample that we synthesize. That's the hardest part, because it's a repetitive process, and we have to keep on doing it and looking at the IR to make sure we're getting the pure sample.
So are there, I guess, any goals for future work that you are considering for this project, or that maybe Dr. Truffin will have in mind for it?
Yes, so the main focus right now is to just purify the compounds that I have, since they're not as pure. So we're just trying to get as pure ball type phthalocyanine as we can, because as I mentioned, the application for these ball type phthalocyanines, there's a wide range of applications. So if we get these compounds to be pure, there will be more research available for other applications, for example, for photodynamic therapy. But right now, I purified mostly of my cobalt chloride compound. I would like to go back to copper chloride and purify them more.
Wow, that sounds awesome.
Yeah.
This was a really great presentation, Riyasa. Good job.
Thank you. Thank you for listening. Thank you for coming.
Thank you. I think someone new walked in or came into the meeting.
Yes. Hi, Ghazal.
Hey, Riyasa.
Hi. So I'll go over my research again. So my research is in inorganic chemistry for the synthesis and characterization of new ball type phthalocyanines. To give you an overview of what phthalocyanine compounds are, phthalocyanine compounds are macromolecule compounds with optical, electronic, catalytic, and structural properties, and they're the second most important class of colorant. Traditionally, they were used as blue and green pigments for automotive paints and printing inks, and as blue cyan dyes for textile and paper. And in the visible region for the electromagnetic spectrum, thiocyanins are limited to blue cyan and green colors. The thiocyanins that I got from my lab were dark green in color. So that's the primary focus for the color of thiocyanins for me. And thiocyanins have a wide range of applications in biomedical and material sciences. For example, in photodynamic therapy for chemical sensors, optical data storage, reverse saturable absorbers, solar screens, laser techniques, and a lot more applications. Ball-type thiocyanins specifically, they are fairly new. They were reported. They were firstly reported in early 2000s. Therefore, there's a lot of research interest in these compounds, and in the lab, there was two-step process that I took to get the ball-type thiocyanins. The first one was to make the monomer, and the second one was to use the monomer and a type of metal to make the polymer, which is the ball-type thiocyanin. As you can see under synthesis, the first reaction is making up the monomer. The second reaction is using the monomer and copper chloride to make the ball-type thiocyanin. And the third reaction is using the monomer and using a different metal, which is the cobalt chloride, to make the ball-type thiocyanin. These were done in a microwave reactor, and the color that I got was very dark green in color. And in order to characterize these compounds, I used IR and UV-Vis spectroscopy. And under characterization, in figure 2, you can see the comparison of the IR spectrum of the ball type that I got from copper chloride, which is in purple, and the monomer, which is in red. So the main peak that we're looking at is this 2235 peak, which is the peak for C2N triple bond, which is seen in the monomer. So essentially, we want the monomer to be consumed in the reaction. So this peak shouldn't be there at all whenever we get the ball-type thiocyanin. So the most pure sample that I've gotten is the sample in figure 2. As you can see, the ball type for cobalt chloride, the 2235 peak is very little. It's not as intense. So that means the sample is not as pure, but it's getting there. And we can also see the emergence of these peaks over here in purple. That is the emergence of C2N double bond in the ball type structure. And in order to purify these, we used TLC and separation to purify the compound and then characterize them using IR. And the third figure shows us the UV visible spectroscopy of three different ball type thiocyanins with 3 different metals. The first one is the copper chloride, and then cobalt chloride and zinc acetate. We can see the location of these 2 peaks are very similar. That is because of the general structure of the ball type thiocyanins. It's because of the different metals used. That was the reason why there's a slight shift in these peaks, but we know that the general structure and the locations should be in the same region. In the future, I'll be purifying my compounds more and also try to grow a single crystal to make sure the structure that I got from the reaction is correct. Do you have any questions?
Great job. I mean, it was not easy, right?
The purifying part was not that easy.
Yeah, I guess. She did a great job. I'm still trying to understand most of the things you said. Like,
yeah, if you have any questions, feel free to ask or just put it in the chat.
Awesome. Thank you. We did such a great job.
You're welcome. Thank you. Thank you for coming. Hi, for the people who just came in, my name is Riyasa, and my research is in inorganic chemistry for synthesis and characterization of new ball type thiocyanins. Thiocyanins are macromolecular compounds with optical, electronic, catalytic, and structural properties, and they're the second most important class of colorant. Specifically, copper thiocyanine is the single largest volume of colorants sold, and traditionally they're used as blue and green pigments for automotive paints and printing inks, and as blue cyan dyes for textiles and paper. And in the visible region in the electromagnetic spectrum, thiocyanins are limited to blue cyan and green pigments. In my research specifically, the samples that I got were very dark green in color. So that is the main focus of my research, to find dark green color compounds. And thiocyanins have a wide range of applications in biomedical and material sciences. For example, for photodynamic therapy, specifically for skin cancer, and for chemical sensors, optical data storage, reverse saturable absorbers, solar screens, laser techniques, and many other applications. The new ball type thiocyanins, which are the large molecules you can see in the second reaction, those were found very recently in early 2000s. So it's very new, and there's a lot of research interest in these ball type thiocyanins because they're reported to be thermally stable and chemically stable than other photosensitizing agent for photodynamic therapy. And that is the reason why these might be a lot less harmful than other treatments for skin cancer. But there is a lot more research going on in that. However, moving on to the synthesis part, the way I synthesized my ball type thiocyanin was a two-step process. The first was to make a monomer, and then the second was to use the monomer and a type of metal to make the ball type thiocyanin. Under synthesis, the first reaction you can see is the making of monomer, and then the second reaction is use of monomer and a use of metal, which is the copper chloride, in a microwave reactor to make the ball type thiocyanin. And then the third reaction is the use of the monomer and the use of another type of metal, which is cobalt chloride, to make the ball type thiocyanin. And both of these samples for ball type thiocyanin were very dark green in color. And in order to characterize these, I used IR and UV vis spectroscopy. Under characterization, in figure 2, you can see the comparison of IR spectrum of the ball type thiocyanin that I got, which is in purple, and the monomer, which is in red. So the main peaks that we're looking for is the 2235 peak, which is the C2N triple bond, which is shown in the monomer. So essentially what we want is the consumption of all the monomers so that we don't see 2235 peak at all in the IR spectrum, because we want our sample to be as pure as possible. And the most pure I've gotten is, this sample, specific sample for the IR. You can see the in purple, the peak for 2235, it's not as intense, it's very little. I'm still in the process of purifying it so that I don't see the peak at all, but it is the most pure that I've gotten. And other peaks that we look for is the peak around here, the emergence of the peaks around here, which is in purple. Those are the peaks that are formed in the ball type thiocyanin, the C2N double bond peaks. So these peaks are the ones that actually tells us, oh, we're getting the correct structure for the ball type thiocyanins. And in order to purify these, I used TLC and separation using TLC. So it's a repetitive process of purifying it and taking the IR, making sure monomer has been consumed, and then purifying it again to make sure the peak is not seen at all. And the second characterization technique that I used was the UV visible spectroscopy. In figure 3, you can see the UV visible spectroscopy with the ball type thiocyanin with 3 different metals. The blue one is with the copper chloride, the pink one is with the cobalt chloride, and the purple is with the zinc acetate. The zinc acetate sample was from past student. I just wanted to compare them with my sample. But as you can see, the location of these 2 peaks are very similar. This indicates that the general structure of ball type thiocyanins is very similar, because all of the ball type thiocyanins, the structures are very same. It's just the metal which is residing in between these complexes that are different because of the different metals that we use. And also the shift in these peaks is also because of the metals. and the intensity depends on the purity and concentration of the sample that I had. And for future, I will be purifying my samples more and using different methods of characterizations other than IR and UV vis. And also after purifying it, I will try to grow a single crystal so that I can actually make sure the structure that I got in the lab is correct. And yes, that was
Nice.
my research.
Nice.
Thank you. Do you have any questions?
I have one. So the peak on this last graph on the right,
Yes.
does the higher the peak equals intensity? Is that what I'm to assume?
Yes. The
Okay.
more higher and the more narrow the peak is,
Oh, okay.
that's the more intense.
Okay.
In my
very
case,
good.
I don't want to see that peak at all. That's why I have to keep on purifying those peaks.
I got you. I got you. And what are your plans to can you tell us? Is it a secret? What are you gonna try first in terms of purification? Or do you have an idea?
Yes. So we're using the same process to purify these compounds, which is using the big TLCs. So what we do is take our sample and just make a line of that sample. And since my sample is very dark green in color, you can visibly see where the sample is. And as we put it in the TLC, the samples move up. And after the TLC is done, when we look at the plate under the UV, you will see some clearless stuff moving up. That is the stuff that we don't need at all because my compound is dark green and I will extract only the dark green line that's left. And other than that, the clearless stuff I don't need because those can be the monomers or the other stuff that is not the ball type phthalocyanine that I need. So that's like a repetitive process of purifying it. So I will continue to extract just the dark green compound and then check with the IR and then do the TLC again.
Okay, great. Thank you. Anybody else have a question?
I think there was a question.
Oh, in the chat.
Yeah. What sparked interest in doing this specific topic of research? So to be honest, when I got into the research with Dr. Trufin, I was not sure what I was doing. I was a little confused because I did read the research papers, but actually doing the experiment myself is different. So I was a lot confused. But when I started learning more about phthalocyanines and the applications and, like using the machines more and looking at the IRs, it got a lot more interesting. And yes, it's a repetitive process. Sometimes it can get frustrating if I don't get any results, but it's just initially I'm getting there. So it's just building up on the knowledge that I've gotten until now.
Good answer.
Thank you so much for listening.
I'm going to leave you right now. Thank you so much for presenting.
Thank
and
you.
At some point, I'm going to ask you to send me this video, okay?
Yes.
Thank you.
Thank you. Thank you for coming, guys. Thank you.
Hey.
Hey, how's your presentation?
So normally, I'm just waiting on people to show up. Only like 6 people show up today.
Oh, really? I've had
Dr.
so many people showing up. Like, my
Really?
throat is so dry right now because, see, I'm presenting to some people and then some other come in and then I have to go over that again. And it's
Yes.
like a nonstop. I just got a break right now.
Okay. I just wanted to check up on you. I'm like, oh, I wonder how she's doing. Did Dr. Shrestha checked in?
Yeah, she's been checking in.
Yeah, same here.
Yeah, but it's going okay actually. I was nervous, but now it's like
I
I'm
think
just
what's
not talk-
helping is like repeating it over and
Right.
over.
Yeah,
and
I'm
over.
getting better at this. Like
So I'm
every
at the
time I
point where I
present.
don't have to look at my notes. I'm just like, I got this.
Yeah, I'm just looking at the applications because I'm just reading it out. But the rest, like the charts and everything, I don't have to
It's
look at
flowing.
anything.
I
Yeah,
know.
it's like this is good, but we're almost there. We only have like what 10 minutes, 15 minutes left.
Yeah, thank goodness.
I know,
I'm
this went by
done.
really fast. I know.
I'm tired. I am done. I am sleep deprived.
I know, but we're almost there.
All right. Well, I'm gonna let you go so you can rest. I just wanted to check up on you.
Yeah, how are your presentations?
So far, people are enjoying it. They're finding it very interesting, and I keep ending it with like, because they ask me questions about like purification and like
Mhm.
any difficulties I experienced, and I told them that the biggest issue was purification.
Yeah, that's what I've been saying that too.
Purification is the biggest issue, and also replicating the synthesis.
Oh yes, because
Mhm.
every time I did my sorry,
That's fine.
I've been talking too much. But every time I did my monomer, the IR would come slightly different than the monomer from before, and it's the same thing that I did.
Did yours change color by any chance? Because it's
For
supposed to be
mine-
white, right?
Mine was white too, both the times that I made them.
Mine was like beige at one point, and then white, and then like I don't know, it was random. And then at one point it turned green.
I think I used your green sample the first time that I did it. You still had the monomer, so
Uh-huh.
I used your monomer, and it was like very light green.
Yes.
But
I was
when
like,
I made
what
my own
happened?
monomer, yeah, but when I made my own monomer, both the times it was like white, so we counted it as colorless.
Mm-hmm.
But it was
Like
weird.
mine had an impurity. I'm pretty sure I used a dirty flask or something. Because
Yeah, the flasks are so dirty there.
Mhm. Yeah.
I put a whole bunch in the acid wash so that I can have a clean one next time.
I think the issue, because I tried doing the reflux apparatus and that didn't work. And so I just did everything on the microwave.
Oh, really? So the first time I did it, the reflux condensation worked so much better for me. So I've been doing the reflux condensation for the monomers because that's what's giving me the
To
white
keep it
compound.
consistent.
Yeah.
Mm-hmm. All right, that's cool. Are you gonna do her research next year?
Yeah. Because I need 3 more hours to keep my I need 15 hours of credit for my scholarship.
Mm-hmm.
So I don't want to take a new class since I already have like very heavy classes next semester. So I asked Dr. Triffin if I can just enroll in her research again.
Yeah.
Because I already know the material and I just need to purify my compound. So I think it'll be fine.
And you get to do more stuff.
Right? Yeah.
Mm-hmm.
And I want to present at like GSIF or something for next semester.
What is that?
It's like
a bigger
it's an
conference?
event from Scholars Academy. Yeah. And some presentations can be and I really want to do the actual poster presentation in person. So hopefully that will be in person.
Okay. That sounds fun.
Yep.
Well, I'll let you go.
Okay. Good luck
Okay.
on the rest of the time we have.
You too.
Bye.
Bye.
Thanks for coming. Thanks for dropping by.
No problem.
All right.